Hello, my name is Marzieh. I'm a PhD candidate at Boston University in bioinformatics. I work in Gary Benson's lab, and the title of my poster is Population-Wide Study of VNTRs in the Human Genome. So I'm going to talk to you about the first ever done high throughput. We took all these VNTR loci on the reference genome and we genotyped them in over 2,500 individuals. So just a brief introduction. What are mini satellites or tandem repeats? I will be referring to them as tandem repeats or TRs. As the name suggests, they are patterns of 7 base pair or larger that are repeated next to each other. So a mini satellite loci can be defined by its location in the reference genome, the pattern consensus, and the copy number. Note that these patterns, these repeats, do not have to be exact and do not have to be complete. A variable number tandem repeat, or a VNTR, is a mini satellite or a tandem repeat that has different copy numbers in a population. When we refer to a VNTR allele, we actually are referring to the copy number, which is an integer number starting from 2, 3, 4 up to any number. So why do we want to study VNTRs? There was a very nice paper in 2019 from the Eichler lab. They showed that VNTRs that humans gained in comparison to primates, apes and monkeys, may play a role in cognitive abilities that we gained in comparison to them, especially the ability to use language. And the ones that we lost are associated with genes related to sensory abilities, such as our ability to taste different food. There has also been many diseases and phenotypes associated to VNTRs. Many neurodegenerative diseases have been associated, such as Alzheimer's, ADHD, Huntington, and also different disorders such as depression, anxiety, and even behavioral traits. Various types of cancers have been associated with VNTRs, and also longevity in different populations of centenarians have been found to be associated with VNTRs. To do this study, we downloaded this awesome data that was generated at New York Genome Center. It consists of 2,504 individuals. None of them are related. Most of these overlap with the 1,000 Genomes Phase 3 individuals, if you're familiar with the 1,000 Genomes Project. They come from the 26 populations and 5 superpopulations. So what makes this data special is that the sequencing was done at high coverage, and they have long read lengths, 150 base pairs, which makes us be able to do this genotyping. We also got the reference TR loci from Tandem Repeat Database, TRDB, and we have a little over 191,000 TR loci. So I want to point this out that these loci that we're looking at is a little over 0.5% of the genome, which is a very little amount. But when you look at where are they located, what is the annotation of this loci, 63% of TR loci are inside genes. About half of the genes in our genome have at least one TR in them. There is some significant enrichment of TR loci inside genes and transcription factor binding sites, suggesting that probably they play a role in regulating genes. So I will go on and talk about my results now. So first, we identified common VNTRs. We define common VNTRs as a loci with different copy numbers in at least 5% of the population. We found 5,699 of them. And then we performed a principal component analysis to see how they cluster, colored them by the population, and we observed that the different populations separate very nicely with these common VNTRs. I'm talking about figure 2 here. Then we saw that, so if these common VNTRs can separate populations, can we identify population-specific VNTR alleles? And we applied a one-side Fisher-Exact test, and we found VNTR alleles that are overrepresented in different populations. We found over 1,200 alleles that were population-specific. and we present them in this virtual gel that I call them, which on the x-axis we have the VNTR loci, and on the y-axis we have the different alleles. And you can see for each population here, African American, East Asian, European, and South Asian, each band represents an allele, and the density of it represents how many um, um, uh, people in that population were inside there. And you can clearly see that there's some bands that only occur in one population. Then we went on and used these common VNTRs to see if they can affect the gene expression. I divided them into 4 different categories. The VNTRs that are inside promoter regions of a gene, VNTRs that overlap with an exon of a gene, VNTRs that are inside the introns, and the VNTRs that interrupt the 5' UTR. When I looked at them, we had like hundreds of genes that were significant, but there was 2 different classes of correlation you could find. So the first one was when there's a linear relationship that as you have an allele, in this case, this is IRF5, the gene, and there's a VNTR in the 3', and the isoform D, when you look at it, when we have the 0 allele, which means the same as a reference, you see that there's a higher expression very significantly than when that is not that allele is not present. Another case is that when the overall expression doesn't change, but the occurrence of an allele, or actually not having that allele, causes some of the individuals to be turned on or off. So here you can see in another gene, a VNTR that is inside the intron. When we have the reference allele, the expression is very, very constant. But in the case that we do not have the reference allele, you suddenly see some of the individuals with a higher probability, they have a very high expression. This would be very significant in gene expression association studies, and also in medicine when they want to find target genes, to be aware of these population differences and differences in gene expression. I would like to take questions. I don't know how this works later, but thank you.